see my slide?
Yes, we can.
You can see my slide.
Yes.
Okay, thank you very much. Once again, you are welcome to this webinar presentation on today's World Malaria Day being celebrated by the Capstone Group. The global title for this year's Malaria Day is actually Malaria Ends With Us. We are to reinvest, we are to reimagine, and we are to reignite the fight against malaria. Here with us now, we are actually focusing on reimagining the strategies in the implementation of interventions against malaria. And like I said, I'm so excited to be here with you and to support in every way I can to support this project. Yes, I've been introduced. I'm a molecular biologist and biotechnologist, and I've actually worked most of the time at Liverpool School of Tropical Medicine, because some of the high-end malaria research we do, we can't do them in Africa. So what we do is start in Africa and take the samples to Liverpool School of Tropical Medicine. So the important thing in malaria fight, I don't know, my slide is not moving. Oh, okay, yeah. Okay, thank you. So there are actually two broad approaches to fight malaria. It's either we go towards killing all the vectors. The vectors are those that carry the parasite. The vectors, those are the ones we all can see. The mosquitoes that bite you during the day and the night, transferring the parasite from one person to another, and going about to breed around our environment. We have insecticides. I'm talking about more about vector control aspect of elimination and intervention. The other hand is actually to kill all the parasites. If you can kill all the parasites, then there won't be any parasite to be carried around by the vector. Malaria, actually, we have to reimagine elimination of malaria. When the epidemic started in America, they actually were able to, in the cities, they were able to eradicate malaria. What did they do? They used what we call DDT, is an insecticide that's very, very effective. So they sprayed every breeding site, every field, everywhere that the mosquito may be breeding, and they eliminated malaria. So you asked me, why did they stop? Why do we have resurgence in malaria disease? After a while, discovered that the DDT they were using, the half-life is actually very long. That means after using it in the field, the DDT will take longer time to degrade. If we remember that what they call silent spring, a researcher actually went around and looked at the effect of the leftover of the DDT on animal, human, and he discovered that it actually have adverse effect on human beings and livestock. So they have to stop using DDT as area spray for malaria eradication. But then they have to come in and introduce new insecticides. So I'll be talking more about the vector angle than the parasite angle. The Capstone project is actually focusing more on both, but more on the vector. The Capstone project is using environmental control, which is going around the breeding site and see how they can eliminate the vector, and also mechanical control, that is using pesticide-treated nets. So this is just the history and the beginning of how we've fared globally in eradicating malaria. In 2000, we can see here in 2000 that we have high incidence of malaria across the globe. And around this time, they introduced vector control tools, long-lasting insecticide-treated nets with pyrethroids. Pyrethroid insecticide. Why pyrethroids? Pyrethroid insecticide, they discovered that the half-life is really, really very, very fast, unlike DDT. And then it is not really on livestock. So they introduced the long-lasting insecticide net around 2000, and you can see the rapid decrease of malaria incidence. Until around 2010, they started seeing that there is really no changes in the malaria incidence. And then the question was, what is going on? Why? And then what was discovered was that the vector, the anopheles, are becoming smarter by developing resistance mechanism against the insecticide. And by 2012, there is really, you can see in this cycle, there's really no change in the reduction in malaria incidence. In Nigeria, every year, when you open World Malaria Report, it gives me, as a person, kind of shame to always see Nigeria as topping the list every year of malaria incidents, every year of malaria death. And it seems that we're actually doing nothing. And the question is, why? Why are we not really doing what others are doing? So at least put ourselves on the path to eliminating malaria in Nigeria. And I'll tell you the reason why. It's because Nigeria, we don't fund research as we should. We don't fund all this intervention as we should. We don't go out to collect data as we should to know where to apply the intervention we are using. I just brought out a broad view of ways of looking at malaria resistance that is being used all across the globe. Any country that you see that have actually eliminated malaria, this is the part of the path that they go into. So they go to the field, collect these mosquitoes, they test them to see if they are resistant to existing malaria control tools they are using. Once they see that they are resistant, they compare them with the susceptible mosquitoes. And in there, they will see if there is upregulation of the transcript inside the mosquito that are resistant. And then we now go in to validate if truly those enzymes that we saw upregulated are actually the one causing the increase in malaria cases. And in validation, we use several methods, including going to metabolomics. The metabolomics will actually tell us if it is enzyme, tell us the structure of the enzyme, and then we can develop effective insecticides that will be able to eliminate them. This is one of the way funding works. This is what CDC USA did in Peru and Guatemala. I was part of this project. So CDC went to Guatemala and Peru and collected mosquitoes that they discovered that they are resistant to the common vector tools, which is the long-lasting pesticide-treated net. And when we screened, we discovered that these enzymes are upregulated. They brought their samples to Liverpool School of Tropical Medicine, and we assisted them to really validate and let them know that truly it is these two enzymes. So what this is telling us now is that in a country like Peru and Guatemala, you don't give net that has pyrethroid only. They will not work. There are new generation nets being produced that will disrupt the activity of pyrethroid, for instance. We call them PBO. So we have PBO plus pyrethroid net. So in countries like Peru and Guatemala, these are the kind of nets that they will be using. So you can see what research can do in unraveling the resistance that that we are seeing in mosquitoes. Without really going deep into what is happening, we won't be able to unravel state. So what are we actually doing in Nigeria? Personally, in our NGO, we have funded some projects, and they are very interesting projects. So when I came back to Nigeria, I decided to run some of these projects. And also, I joined, this is Professor Oyinbo here at the Andy Center of Malaria Diagnostic Center, where we actually did some parasitology work. And I'll just be talking about some of what our findings and which will be relevant to your project that you are going to be doing at Abuja. Not every long-lasting insecticide net that you see that are active, especially those that are being sold outside what is being distributed by the Ministry of Health. So what we did here was to go to households in Port Harcourt, and we collected dead nets from them. And then we went to the market to buy what is on sale in the market in Lagos. So our NGO is in Port Harcourt, so most of our work are actually done in Port Harcourt. So we discovered that various households got their bed net from several means. But one important thing is that you see most of them are actually only 1 out of 3 got theirs from the hospital. And then you can see 2 out of 3 actually got it from friends. What this implies is that most of the people that are supposed to be distributing the net from Ministry of Health, because on the net, we discovered that the name not to be distributed and the name of Ministry of Health are written boldly on those nets. So how did they get to the hands of other people that are not distributing it as gifts to their friends? It's because those people that are supposed to be distributing this net, they kept them for themselves, and some for 6 years, 8 years. Some of these nets were there, they did not use them. And then we actually checked how many people are really sleeping under the net. In one of the research we did, we discovered that very low percentage of people are actually sleeping under the net. And they will give you several reasons. Some will say when they sleep under the net, they usually have bad dreams. And some will say they can't breathe very well. Whatever excuses anybody is giving, sleeping under net is still the best, especially for those people that are living in the rural area. Malaria is a tropical disease and loves rural area. You can't see much of malaria incidents in urban cities. Like Lagos, there are really very low percentage of malaria still existing in Lagos and from other urban cities. So in this research, we discovered that not many people are sleeping under the net. And then let's see how this net, how they compare with the one we bought in the field. So we did the quantification of the active ingredients in the net. And what we discovered was that even though those nets have been kept for years, some of them still have enough, and some of them have been using the net actually for like two years. The expiry, really there's no expiry date on net, but most of the time it's around 3.5 years that you are advised to use the net. After washing for like 20 times, you are supposed to change it. So after several washing, we discovered that most of the households are still having some of the active ingredients here. So in the first one is 0.3, second 0.6. But what is interesting is look at the one we bought from the pharmaceutical store in Lagos. It has 1.5 based on our quantification. The standard should be 1.8. But similar net that we collected from the field is actually having 0.67 as the concentration we quantified. And this is less than half of what we discovered that was in the one we bought in the field. And then when we did CONASY and we matched the quantity with the CONASY is the way we test for efficacy of bed net. So we put mosquitoes in there After 5 minutes, we remove them and we begin to watch them as when they will be knocked down. KG there is knocked down. And then after 24 hours, we count those that are dead and those that are alive. So when we did the cone assay, I'm particularly interested in these two. You can see, yes, all of them attain similar knockdown percentages. But the net that has 0.6, less than half of what the one we bought in the market had, actually did better than the one that are being sold in the market for people. So what explanation can we give? It is very simple. This is actually, I would say, the first experiment that will be clearly relating quantification to efficacy. So what we discovered, one, was that the net being sold in Nigeria, they call it permanent one. There are actually no permanent one being released by WHO. Permanent one was the first net the company made, and it did not meet the standard. So the people making this net for them, usually most of these production are done in, let's say, China and Co. They know the quantification, they know everything, so they continue to use it. But permanent 2.0 that they made, they've actually done something to the way they impregnated the net, such that the release rate is even across the 3.5 lifespan of the net. That was what they could not get in the net that they are selling, that are fake in the market. So this is telling us that it's not just any net, because I read in a paper that Nigeria is about to start making their own pesticide-treated net. Yes, we can do it, but it's not just about putting the insecticide in the net. It's about the technology to evenly release those insecticides so that they will be able to kill the mosquito. Why is it important to effectively kill the mosquito? Once you begin to expose these mosquitoes to insecticide like this and they're not dead, that's what we call selection pressure. Selection pressure, over time, the progeny of these mosquitoes will develop resistance. And over time, you have more of them not being killed by the insecticide we are using. So it is very important when you are getting your net, get it from the right source, and then make sure that they are one approved by WHO. Yes, we need to test if we are to end malaria in Nigeria. This is one thing that we all are guilty of. Once we think we have malaria fever, we just go to, because it's OTC, you just go and buy malaria drug. You can raise up your hand if you have done that. I've done that as well. But then it is not good. You must be tested before you go out and take malaria drug. And we have RDT malaria kits that are very effective. These were part of the project we did at Andy Center in Unilag. We actually compared 5 RDTs. Some of the RDT can detect across all malaria parasites, because we have like 4 that are malaria parasites that infect man, and some of them can detect across board. But the most important malaria parasite in Nigeria is falciparum, and all these, anyone you pick will actually be able to detect malaria parasite. But the gold standard of testing malaria is microscopy test. Why? Because the microscope will be able to tell you the stages of those parasites in the blood. There are some drugs that will work depending on the stage of the parasite. So when your bloodstream, your blood cells are being monitored on microscope, you'll be able to see the levels. They will be able to do the counting and tell you the parasomia level in your blood, and also the type of the parasite and the stages of the parasite. But one thing that we discovered in the field was that not every microscopist are well trained to actually be able to read the microscope. Most of the time, what they look at and they thought was parasite, most of the time are not parasites. We've done research based on that, and we saw that most of the time they're wrong. And the problem with that also is that if you are using malaria drug without testing if the parasite in it, there will be residual in your blood, and when the parasite gets in, it can begin to develop resistance. We all remember chloroquine. Some of us can chew chloroquine as bitter as it is. You can chew it, you got used to it. And they have to remove it because they discovered that the parasites have started developing resistance to heat. So now we have a new malaria drug that we can use. Keeping chloroquine for when those ones, maybe they will fail one day, and also continue to develop very effective malaria drug as we move on. So to help in detecting correctly the malaria parasite, there is a company in South Korea that developed this AI-enabled digital microscopic test. And this was the lab, Andy lab in Unilag we were testing. This was what I was actually looking at here. And this will take off any human error or subjectivity to detecting and detection. So this machine has been trained to detect, to do the counting. This result is from the machine. And it will tell you within 5 to 10 minutes whether you have malaria and the type, and also tell you the stages of those parasites. So in carrying out your project, I think this is, in terms of cost, definitely the machine is very expensive. So we still have to rely on RVT test, because the only difference with RVT, or one of the disadvantages I would say with RVT, is that if the antigen, even if the malaria parasites, if they are out there, if the antigen is still there, the RVT will detect it. But then if it actually detected parasites in your body, that means you've been exposed to it, maybe it's still there or not. When you use malaria drug, it will still be better testing than not testing and just going ahead to use malaria drug. Also, we need to identify and locate properly breeding sites all around us, especially in FCT. It is very important to know where they breed and also to eliminate those breeding sites. These are some of what we did in Lagos here, and some of what I did in Cameroon and Benin also, looking for breeding sites. It's dirty work, but these are part of sacrifices scientists we have to make to help in eliminating malaria if we have enough funding. There is no intervention we are doing that will be effective if we do not create awareness, if we do not tell people that these are the means and ways to prevent and eliminate malaria. In the last presentation I did, I promised to bring up some of the budget estimates that we use in Port Harcourt. So this is one of them, and depending on location, I know Abuja is expensive, so you may expect to see the cost going higher. But then you need to talk to the community leaders. Very important. And sometimes they won't listen to you until you talk to them in a way they will accept you. We all understand what's going on in Nigeria. And then even when you call people and you don't give them anything, the next day you ask them to come, they will not come. If you gather them and you give them ordinary zobo, next time they will be the one inviting more than 1, 2, 3, 10 people to come with you. So if you are going to be organizing awareness programs, make sure you come. The best is give them jollof rice. If you give them jollof rice, you can be sure all the people in that locality will turn up. the next day. But then it's not about the cost, it's about us educating people. You won't believe it. One of the projects we did in Port Harcourt, so many people have not even heard about mosquito net. In fact, when you give them mosquito net in rivers, you know what they do with it? They will take it to the river and be fishing with it. And believe that you help them in their business, whereas they're supposed to be sleeping under the net. Some pregnant women, they don't know that um, there are public health centers around them that the federal government has actually paid to treat them against malaria. There are a lot of programs, a lot of therapy dedicated for pregnant women, for children under 5. And now we also have the vaccine. How would they know if they're not told? So educating them is very important, and creating the awareness is also very important. Another way to get them is to go to the biggest church in the locality. Once you talk to the pastor, and then you come with anything for them, even if it's paracetamol, say you're doing health week or whatever, they will listen to you. In Nigeria, nothing goes for nothing. If you must get them, you must be ready to spend money. So I want to say this is one of the key things you need to map out to know how much you'll be spending. In Abuja, you probably be looking at getting halls and then getting a better PA. But in Port Harcourt, because it's, where we go to is a bit rural, so we can go with just spend 400,000 and then we get things done. I've come to the end of my presentation, and I would like to wish Public Health Capstone Group SPPG class of 2025 great success in tackling malaria in Nigeria. Beyond the bite shouldn't just be a slogan. It should be what we all should imbibe, even if you are not suffering from malaria. In our malaria day presentation, I actually told people that I was a survivor of severe malaria, and I still carry on the long-term effects of that malaria. It's something I won't wish for anybody to, especially people that use brain like me, to have. So we must end malaria in Nigeria. We must, and yes, it's everybody, all hands must be on deck. And it ends with us taking a position, forcing the government to have political will to want to do, showing them data, even if they're not sponsoring anything, we should do. And I'm so glad that you guys are going to be collecting data, analyzing data, and also sharing your data for people to see. Yes, we can end malaria in Nigeria if all of us agree to do so. Yes, we can. Thank you very much. Thank you for listening. If there are questions, I'm available to answer them. Thank you very much.
All right. Wow.